In the complex world of metastatic diseases, understanding events and tropisms is crucial. Welcome to the tutorial on the MSK-MET analysis using the code interpreter feature of ChatGPT. The data set taken for this analysis is from the CBio portal. So I click on this link, it navigates to the corresponding bio portal. So which is for Cancer Genomics, and the data set that I have taken for the current analysis is a Memorial Sloan Kettering Metastatic Events and Tropisms dataset. It is an integrated pan-cancer cohort of tumor genomics and clinical outcome data of 25,000 patients. So click on this download link. You will get your database being downloaded, which contains 25,775 samples. And the file type is TSV type. So I have gone to the ChatGPT, the code interpreter. So in this code interpreter, I have clicked this plus link in order to upload the corresponding file. And the first thing that I had given, the first question I had given for the ChatGPT is to perform the statistical analysis on the data set. Once I press enter, it came out with the following results. So it has loaded the data set and it has given me the Python code which has worked on the data set, and this shows the interpretation about the findings. So it shows that the data set is a clinical data which is related to the patients with various types of cancer, and it has drawn the inferences about some of the columns, like we have identifier, which is the study ID or the patient ID or the sample ID, the age information, and the cancer information, that is the cancer type, its subtype, etc., the sample information, demographic data, and the genomic information. So it has asked me whether I am interested in what type of potential analysis. So do I want to know the descriptive statistics or distribution of the cancer types and subtypes in the data set, or correlation between the various numerical variables, or I want to compare the different groups like tumor purity based on the cancer type or the sex. So now I give all the analysis as my reply to this particular chat. So it has broken down this one into the various topics like descriptive statistics, distribution analysis, the correlation analysis, and the group comparison. So for each of this one, the Python code has been generated and it gives me the interpretation for each of the results that it has drawn. So it has specified the age related metrics. So age at the death of the patients were in the range of 19 to 90 years, and the average age is 64.9, and the age at first mets is nothing but the patients who were first diagnosed with metastasis. The average age of the patients were 61 years, and age at sequencing was found to be around 62.3 years. So age at the surgical procedure is approximately 61.5 years. The genomic metrics it has drawn inferences are the fractional genome altered, MET count, MSI score, that is the microsatellite instability score, the mutation count, and the tumor mutational burden average value. Similarly, the sample coverage and the tumor purity has been given. This is followed by the visualization of the categorical variables like cancer type, its subtype, the sample type, and the race category. So it has provided the Python code and the various distributions. With respect to each of the data distribution, it has drawn the inferences. So the cancer types that has been found in the data set are the most prevalent are the breast cancer, endometrial cancer, and colorectal cancer. The distribution of the sample types or metastatic samples are predominant in this particular case. The race category is we have the Asian, Far East or Indian subcontinent, and the black African Americans are there. Distribution of the cancer subtypes is nothing but like breast ductal HR plus HER2, and the colorectal adenocarcinoma or being the common one that has been explored. Then it has given me the Python code to understand the correlation or the relationship between the numerical variable and it has drawn the inferences about the strong correlation that exists between FGA, the age at the deaths, and age at first meds and last contact, and the sequencing and the surgical procedure, the moderate correlation that exists between the TMB and med counts, the weak correlation. Most of the other variables seem to have the weak correlation in this particular case, and it has also compared the tumor purity on the cancer types and the corresponding gender. So it has given the various plots related to that one. So it has interpreted that the tumor purity based on the cancer type. So if it's a breast cancer, it tends to have a higher median tumor purity than compared to that of the endometrial cancer or colorectal cancer. The spread of the tumor purity is also different for different cancer type. About the tumor purity with respect to the gender, it has given the interpretation that the male and the female have the similar distribution of tumor purity. There is no significant difference in the median value. So then I have asked to apply the relevant machine learning model on the data set, but I have to define a very clear objective related to what is my, if it is a classification problem, I have to specify my target variable. If it's a clustering type, I have to specify what are the clusters that I want to identify within the distinct subgroups of the data. If it's a regression, I have to take the continuous feature as my dependent variable in this particular case. The variable which is continuous in nature and also numerical has to be taken into account as a different variable. So it has given me the potential ways by which I can model my machine learning model on the particular data set. So if it's a classification, it can predict the cancer type, and it can also predict the sample type. If it's a regression type, it can predict the tumor purity or the age at the death based on the diagnostic or the tumor or the genomic feature. And for clustering, it is going to group the patients into cluster based on the features to identify the distinct subgroups within the data set. So I have first asked it to predict the tumor purity, which is a regression model. So it has prepared the data, it has performed the model selection, and it has trained and evaluated the model. Since it is the regression analysis, it has produced the performance metrics for the regression analysis, which is based on the mean absolute error and the R squared value over here. So it has specified that the linear regression model with a selected feature might not be the best fit for this kind of prediction in this particular case, but we need to enhance this one by providing the additional features in this particular case. So I have moved on to the prediction of the cancer type used on AutoML, but since it couldn't work with the AutoML, it has used TPOT, the corresponding library, but it could not work with this particular TPOT because it came out with a particular error. So it has suggested me whether it has to work on a random forest or the gradient boosting. So when I gave yes, it has proceeded with the random forest, and it has implemented with the corresponding random forest classification algorithm. Again there was an error, but it was able to correct the error in this particular case, and it came out with the corresponding accuracy score, which is also very low. So it has suggested me to to work on more advanced model, perform the feature engineering or more comprehensive hyperparameter tuning in order to provide the better performance.